Welcome to episode 8 of our series on creationism versus evolution, the origin of life, a non-existent probability. In today's episode, we'll delve into the fascinating journey from chemical compounds to the complex structures of life. It's often stated in contemporary theories that through chemical reactions, powered by electrical currents like lightning, in conjunction with external conditions such as atmospheric pressure, the building blocks of life were formed. This foundational concept in the science of life's origin was established before we even had a clear understanding of what a living cell is and how it's structured. For decades, scholars and intellectuals were educated to believe in the spontaneous generation of life, a belief that persisted even after the true nature of the living cell was discovered. The theory adapted itself seamlessly to new findings, maintaining its relevance. Consider the living cell, the most complex system in the universe, not just complex, but the epitome of complexity, surpassing anything created by nature or human hands. Despite our modern tools and astounding knowledge, replicating the spontaneous formation of a living cell remains beyond our capabilities. Decades of designed experiments and manipulations cannot mimic the random processes that purportedly led to life. The simplest living cell, we're told, has a mechanism far more complex than any machine ever conceived by humans. This perspective echoes in the words of Thorpe W.R. Bird, highlighting the cell's unparalleled complexity. Fred Hoyle famously analogized in Nature, 1981, that the likelihood of higher life forms arising in such a manner is comparable to a tornado assembling a Boeing 747 from the materials within a junkyard. A compelling metaphor for the improbability of spontaneous life formation. It's crucial to clarify that the origins of life are not an integral part of evolutionary theory, which focuses on the development of species. Abiogenesis, the theory of life's spontaneous origin, serves as a precursor theory explaining life's initial emergence. Exploring the composition of the cell, we find it consists of proteins, which are sequences of amino acids linked by chemical bonds. The precision required for a functional protein is staggering, with a single incorrect placement rendering the entire structure dysfunctional. The probability of a single protein forming by chance is astronomically low, akin to a monkey typing out the history of humanity on a typewriter without error. The improbability extends to the formation of the first cell, with geological perspectives suggesting it's so unlikely as to be virtually impossible across billions of years and planets. This skepticism is shared by many in the scientific community, pointing to a requirement for design and purpose behind the formation of life. Yet the cell is more than just proteins. It includes nucleic acids, carbohydrates, lipids, vitamins, electrolytes, and more, all functioning in concert to enable life. Without this orchestrated functionality, life cannot sustain or reproduce. The very first cell, in isolation, offers no advantage unless it can replicate, which involves a complex interaction of enzymes and genetic codes. Moreover, the chirality of amino acids presents another layer of complexity. Proteins within living cells exclusively use left-handed amino acids, despite the natural occurrence of both left and right-handed forms. The specificity required for life contradicts the randomness assumed by theories of spontaneous origin. In the end, the statistical improbability, combined with the intricate design and function of even the simplest living organisms, leads some to argue for intelligent design as the most plausible explanation for the origin of life. This view suggests that the complexities and specific requirements for life exceed the capabilities of random processes, pointing towards a directed intelligence behind the existence of life. Now let's continue diving into the 1953 experiment by Stanley Miller, a cornerstone in evolutionary teaching. Miller's experiment aimed to simulate early Earth's conditions by mixing ammonia, methane, hydrogen, and water vapor, then heating this mixture to 100 degrees Celsius and zapping it with electricity to mimic lightning. This process produced 3 out of the 20 amino acids found in proteins, sparking significant excitement in the scientific community. However, critiques of Miller's experiment highlight that it might not accurately reflect natural conditions for several reasons. Firstly, Miller used a cold trap to isolate the reaction's products from the experimental heat, a condition unlikely replicated in nature. Secondly, it's now believed that methane and ammonia were not prevalent in Earth's early atmosphere, Thirdly, the high levels of oxygen present would have made the formation of amino acids in such an environment impossible, as methane would decompose into carbon dioxide and water, and ammonia into nitrogen and water. Furthermore, without oxygen, there would be no ozone layer to protect emerging life forms from harmful ultraviolet radiation. Despite the foundational status of Miller's experiment in evolutionary biology, the conditions he simulated are now considered by many to be quite different from those of early Earth. This has led scientists to propose that early Earth's atmosphere consisted mainly of carbon dioxide and nitrogen, which are less reactive than the gases used by Miller. This discrepancy raises significant challenges for the spontaneous formation of life's building blocks. Moreover, the DNA within every cell, composed of various sequences of 4 nucleotides, stores the blueprint for the organism's entire structure and function. The complexity encoded within DNA is so vast that if it were to be written out, it would fill a library of 900 volumes, each 500 pages long. Yet, this immense information is compactly stored within the microscopic nucleus of a cell. The chances of a single gene forming randomly, necessary for creating a simple protein, are astronomically low, leading some scientists to describe the origin of life on Earth as almost a miracle. This immense complexity has led to skepticism about the likelihood of life arising spontaneously from simple organic compounds. In response to these statistical improbabilities, some scientists, including Nobel laureates, have explored alternative theories like panspermia, suggesting life might have originated elsewhere in the universe. This perspective underscores the challenges and mysteries still surrounding the origin of life, suggesting that our understanding is far from complete. Richard Dawkins offers an original solution to statistical problems. Let's assume, according to our best guess, that the probability of spontaneous appearance is a very small number, say 1 in a billion. Clearly, this probability is too small to give us any hope of repeating such a miraculous event as the appearance of life in our laboratory experiments. But if we assume, and we are certainly entitled to assume for the sake of argument, that life appeared only once in the entire universe, it follows that we are allowed to incorporate a very large amount of luck into the theory, because there are so many planets in the universe where life could have appeared. If there are 100 billion such planets, as one estimate claims, then this number is 100 billion times greater than that probability of spontaneous occurrence, from the blind watchmaker P160. Let's grant Dawkins the benefit of the doubt in his generous theoretical number of life appearing at 1 in a billion, and set aside the rest of the development whose chances are far less than the number of atoms in the universe. Let's focus on his main argument, reminiscent of the multiple universes argument and no less foolish than it. What is the significance of all the galaxies in the world when we have no access to them? We haven't checked, and of course we cannot check all the other galaxies in the universe, so they cannot be included in the statistical calculation. According to this approach, we could dismiss all research and experiments in reality. An experiment conducted on 5 mice is meaningless because there are, for the sake of argument, 5 billion mice in the world. Hence, we are allowed to incorporate a very large amount of luck into the experiment. The mistake is, of course, that the experiment only covered 5 mice, so all the others that were not tested are irrelevant to our case. We don't know that life didn't develop in other universes, so how do we include them in the statistics? In conclusion, while experiments like Miller's offer intriguing insights into the potential pathways for life's chemical beginnings, the complexity of life, especially the intricacies of DNA, points to a puzzle that is far from solved. The debate between creationism and evolution continues, with each discovery and hypothesis adding depth to our understanding of life's origins. Thank you for joining us on this exploration of one of science's most enduring questions. Stay tuned for more discussions as we delve deeper into the fascinating debate between creationism and evolution.